Hi everyone, Professor Gustin here, and we are on microbial genetics this week. And I have a lovely scanning electron microscope picture of some bacteria, but we all know inside of that bacteria is a single circular chromosome made of DNA. And DNA is the instructions on how to make that bacterial cell and how to conduct all the metabolism and do everything that bacterial cell needs to do. And so we're going to talk a little bit about that DNA and the implications in the field study of genetics. Now, you're probably familiar with the idea that DNA is the hereditary material in the cell. Okay, that's the material that has the instructions on how to make the bacterium. You should also have some experience with the idea of DNA, RNA, and proteins, transcription and translation. I am going to assume that you have some basic knowledge of this. This course, the prerequisites for this course would require you to have some experience in this topic. That being said, I know a lot of people get pretty rusty on this material, and it can be kind of challenging. You might not have even been great at it the last time you learned it. So I highly recommend just doing a little review. Some of my favorite videos are these Khan Academy videos. So there's different ones related to genetics. The one that I'm recommending here, it talks about the central dogma. If you just Google Khan Academy and central dogma, you will find some information. You'll find some other videos related to genetics. So I don't need you to understand in more depth than what I'm going to go over, but you might just need a refresher to remind yourself what the heck is DNA and RNA and proteins and what's transcription and translation. This link is also on your study guide. And like I said, you can just Google Khan Academy and genetics or central dogma. You'll find some information about this. So I'm in section 8.1. And this is just some basic genetic information. It says define the terms chromosome, genome, gene, and compare them in eukaryotic and prokaryotic cells. So remember, bacteria are prokaryotic cells. Most of what you've learned in other classes has probably been about eukaryotic cells. And we're in the process of comparing prokaryotic cells to eukaryotic cells. So when we talk about genome, chromosomes, and genes, it's pretty consistent in eukaryotic cells and prokaryotic cells. When we talk about the genome, we mean all of the genetic material in a cell. So this cell, in this picture, it happens to be a human cell. I can tell because it has these linear human chromosomes. So the genome is all the genetic material or all the DNA in a cell. A chromosome are the individual pieces of DNA, and you might remember human chromosomes are made of human cells. The nucleus of human cells contain 46 chromosomes. And remember, chromosomes are big, long pieces of DNA. It's showing you here how that DNA is all folded up, and now they're stretching it out here. So the genome is all the genetic material in a cell, whether you're a human cell or a bacterial cell. A chromosome are the actual pieces of DNA that contain, that together make up the genome. Human chromosomes, we said there are 46 chromosomes in the nucleus. Bacterial chromosomes are usually one single circular chromosomes, but remember, prokaryotic cells don't have a nucleus. And finally, the concept of a gene. So if we take a chromosome and we stretch out some of this DNA that's in that chromosome, a chunk of that DNA or enough DNA to have the instructions on how to make a protein is called a gene. Enough DNA containing the instructions to make a single protein is what we call a gene. You do not need to memorize these numbers, but I think they just help give you some perspective. So I'm going to compare human cells to E. coli, a typical bacterium. And so the size of the genome, remember we said the genome is all the genetic material. And how do we measure that genetic material? Well, you might recall when you look at DNA, that DNA has base pairs. It has ATs and GCs. You might hear these called the nitrogenous bases, but we have these base pairs. So when we talk about how big is a piece of DNA, we say, how many base pairs does it have? And so, there's 1, 2, 3, 4, 5, 6, 7, like that, okay? So when we look at human cells, there's about 3 billion base pairs, these ATGC pairs in our DNA in every one of our cells. Remember, every cell in a multicellular organism contains all the instructions on how to make every protein in the body. So just because my eyes don't have liver in them, they don't have liver proteins, but they still contain the DNA that has the instructions on how to make the liver proteins. So all the instructions on how to make the whole body is in every one of our cells. So human cells have about 3 billion of these ATGC pairs in the DNA. And E. coli, comparatively, has about 5 million base pairs, ATGC pairs. So E. coli, being a single-celled organism, being a smaller organism than our cells, you can imagine that E. coli probably isn't quite as sophisticated as an E. coli cell is probably not as sophisticated as a human cell. So what it's telling us is it doesn't take as much instructions to make an E. coli as it does to make a human. Not surprising. How many chromosomes? We already said human cells have 46 chromosomes. E. coli only has that one, has one circular chromosome. And then the number of genes, we said the gene is enough DNA to make a protein. So human cells have around 20,000 genes. And so we have 20,000 different proteins, and we probably have more than that because sometimes proteins combine with each other or get modified in certain ways to function normally. But let's say we have about 20,000 genes in a human cell and only about 4,000 in an E. coli cell. So again, you can imagine that it takes fewer different kinds of proteins to run a bacterial cell than it would take to run a human cell. So genome, chromosome, and genes. One of the last things I want to talk about in this section is the concept of a plasmid. So here's that picture of the prokaryotic cell we've looked at before. Here's that single circular chromosome that's all kind of folded up in the cytoplasm there. But you also notice that there's another little circular thing right here, and that's called a plasmid. And what a plasmid is, it's a piece of DNA outside of the chromosome. So here we have the chromosome that has the instructions on how to make all the parts of the bacterial cell, but you've got a little extra DNA in some cells, not all cells, and there can be one plasmid or there could be many, but we've got these one or more extra pieces of DNA that has some extra genetic information. The plasmid are not essential. Where did I go? The instructions in that plasmid are not essential for normal metabolism and growth. I'll say that again. The instructions or the DNA in that plasmid is not essential for normal metabolism and growth. I kind of think about it. If you look at all the DNA in a cell as a recipe book, has all the instructions on how to make everything in the cell. The plasmid has some additional instructions, but they might not be things that you absolutely need for a recipe. Maybe it's got the frosting or some kind of sauce or something. So they're not essential for your recipe to work. But the genes on those plasmids might give the bacterium certain advantages. And things that might, and I think about this as giving the bacteria a superpower. We've heard a lot about antibiotic resistance. that sometimes you can get an infection and an antibiotic that normally killed the bacterial cell no longer kills it because the bacteria became resistant. Well, frequently it's because the bacterium gained a plasmid. It gained some instructions on how to fight off that antibiotic. So it might give the bacteria a resistant factor. So under normal day-to-day activities, the bacteria doesn't need this plasmid until it gets exposed to an antibiotic, and now it can make some kind of protein that will help the bacteria resist that antibiotic. It can also help it be virulent, and it can help it cause disease. So plasmids kind of give bacteria superpower. Not all bacteria have plasmids, and some bacteria that have them have a lot of them. The last thing I wanted to mention about just general background on genomes and genetic vocabulary like this is just to bring in viruses. And so it says, describe the characteristics of the viral genome. Now, remember, we talked about viruses when we talked about acellular pathogens. And we said viruses can have all viruses have some kind of nucleic acid. They have some kind of chemical that contains the instructions on how to make the virus. All eukaryotic cells have DNA. All bacteria have DNA. But viruses can have DNA or RNA. It has one or the other. Okay. So the other thing about viruses and their genetics is they have genomes that are 10 to 10,000 times smaller than bacteria. And the smallest viruses only have about 2 genes. If we go back and we look at our table, we said if we're looking at the genome size, 5 million base pairs in E. coli, and about 4,000 genes, we said viruses can have 10 to, 1,000 times smaller, 10,000 times smaller genomes than bacteria. And some viruses only have 2 genes, and they would be to make specific parts of the virus. So viruses have much smaller genomes. And so my question was, why? Why can viruses function with such small genomes? And I'll go back to an analogy that I probably used earlier in the semester. Remember, viruses are obligate intracellular parasites. Viruses have to live inside a host cell to multiply. And I think about it like kids living with their parents. Kids just can't go walking around. They don't have money. I mean, they need a house, they need food, and they're going to get that from their parents. So the kids are like the virus, and the parents in their home are the host. So the idea is the kids don't need money because the parents have money, usually, and the parents supply the house and supply the food and supply the clothes. So viruses are like the kids living off the parents. The virus doesn't necessarily have to be able to make all sorts of enzymes and all different kinds of metabolic machinery because the host cells already do that. So why can viruses function with such small genomes? Because viruses are obligate intracellular parasites. They live inside a host cell that can provide them with a lot of the needs that they would have. They don't have to make all their own proteins. And that's where I'm going to stop, section 8.1.